Hello, dear students. I welcome you all on behalf of Shri Jendra Rangjun classes. Dear student, we are in a continuation of a chapter that is plant kingdom. And in previous classes, we have already discussed about algae and the different classification system. In today's lecture, we are going to talk about bryophytes. So, bryophytes are somewhat evolved plants compared to the algae. Okay. And if you talk about this algae specifically, this bryophyta, this term bryophyta is proposed by Robert Brown. Robert Brown. Okay. And the study of bryophyte is known as bryology. If we talk about father of bryology, father of biology is Hedwig. Scientist Hedwig is known as father of biology. Some scientists believe Caver, Cavers, Cavers as a father of, according to some scientists. Whereas if we talk about father of Indian biology, then he is Professor Sivraj Kashyap. Okay, so these are the different terminology and the fathers which are associated with the bryophytes. Now we are going to mainly talk about bryophytes, and if we talk, these are somewhat evolved than the algae, and this bryophytes going to include the liverworts, hornworts, and mosses that we are going to discuss in detail. If we talk about habitat of this bryophyte, these bryophytes are commonly growing in moist shaded area. So what you need to remember here, in which habitat they are found, moist shaded area, and specifically in the hills. So due to their presence or occurrence in the shady areas, they are also known as cyophytes. They are also known as cyophytes. Okay. Now, so you can see these are the moist shaded areas where they are found. Now, bryophytes are also considered as archegoniate. Archegoniate, why they are considered as archegoniate? Because archegonium is a female sex organ. In algae, we have studied that the female sex organ is named as oogonium commonly. Now, in bryophytes, female sex organ is known as archegonium. So, these bryophytes are the first archegoniate. These bryophytes are first archegoniates. Embryophytic means they are first embryophytic. In algae, we have studied that zygote undergo meiosis, but in bryophytes, we will study that zygote undergo mitosis and form an embryo, so they are embryophytic. And atracheophytic. Atracheophytic means vascular tissue absent. whereas they are cryptogams means seedless plants. So this is the thing. First tracheophytes, first embryophytes. Embryo was development was first seen in case of the bryophytes. That is the second group in case of plant kingdom. They are atracheophytes. Vascular tissue are absent, and they are, do not possess a seed, so they are a seedless. So then they are also called as amphibians of plant kingdom. They are also called as amphibians of plant kingdom. Now the question is why they are known as like this? Because this plant live in moist soil and are dependent on water for the sexual reproduction. They are dependent on water for sexual reproduction. Actually, male gamete are motile in bryophytes, and this male gamete use water as a medium to reach the archegonia or to reach the female gamete which is present in case of the archegonia. So due to this, they need a water. Because of this, they are known as amphibians of plant kingdom. You can see this is the watery condition in this moist soil. They are known to grow. Here also you can see some of the presence of bryophytes. Now these bryophytes are also considered as primitive land plants. Actually, they are land plants, but they need water for fertilization, as we discussed in the previous slide. They usually occur in damp and shaded places. They usually occur in damp and shaded places, and they are primitive land plant. They are considered as a first land plant or first terrestrial plant. They originated from the aquatic plant actually, and they come on land through the water. So due to their origination in the aquatic plant, some of the characteristics of bryophytes are similar to the aquatic plant. Okay. Then, if we talk about this bryophytes, they play a great role or important role in plant succession or bare rock also. So we will discuss that in a bryophytes. The last group which we are going to discuss, that is mosses. And in mosses, dear students, what happens? They are used in this plant succession or ecological succession. Suppose there is a one barren area where there is a no life, but after years, thousands of years, there will the forest will be seen at that particular location. So on a barren area where there is a no life, initially the lichen will grow, and after lichen, this mosses will grow. And same way, the higher plants keep on growing, keep on growing, keep on growing, and replacing the existing one on the barren rock area, and at last the forest will be developed. So this successive event is known as plant succession. The development of life or plants on a rocky area is succession, and in that also they grow after the lichen. So you can see this is the rocky area. Now the plant body of bryophyte is more differentiated than that of algae, but the plant body is thallus-like. It can be prostrate or erect, means it can be vertical or horizontal. Or prostrate means horizontal and erect means vertical. And attached to the substratum by a unicellular or multicellular rhizoids. So actually they have a thallus-like body, means they have a root-like, stem-like and leaf-like structure. The rhizoids are the root-like structure. Rhizoids are actually root-like structures. So this thallus body is present. But in mosses, higher mosses, you will see the presence of differentiated or developed gametophyte and sporophyte. And you will also see these rhizoids. They can be unicellular or multicellular. When we will study the groups, then that time we will discuss in which the unicellular rhizoids are found and in which group multicellular rhizoids are found. So they lack a true root, stem and leaves, but they have a root-like, stem-like and leaf-like structure. root-like structure is rhizoid, leaf-like structure is phylloid, and step-like structure is known as colloid. So you can see this is the diagram of particular one. This is the root-like structure. These are the leaf-like structure. This is the stem-like structure, and this is forming a gametophyte. And this is a sporophyte, and sporophyte is differentiated into stalk and capsule. It is differentiated into stalk and capsule. So if we talk further, we come to know that the plant body consists of parenchymatous tissue. If we talk about the majority plant body of bryophyte, the plant body is made up of parenchymatous tissue. And if we talk about the plant body of bryophytes, the main plant body of bryophyte is haploid, means that is gametophyte. Which one? Gametophyte. So gametophyte is a haploid body, it is a gamete producing body, and it is a dominant body in case of bryophyte. So this gametophyte is going to produce gametes. So because of this, it is known as gametophyte. And the sex organ in bryophyte are multicellular, jacketed and stalked. In algae, we have studied non-jacketed unicellular sex organ usually. In bryophyte, usually sex organ are multicellular and jacketed and stalked. So this is the antheridium and archegonium, that are the male and female sex organ. It is male sex organ, it is female sex organ. We will discuss about this in detail. So the male sex organ is known as antheridium, female sex organ is known as archegonium, and the antheridium produced biflagellated anthrozoid is one of the important point in case of bryophytes. In bryophytes, dear students, you need to remember that the anthrozoid or male gamete, anthrozoid are nothing but what? They are male gamete. This male gamete are motile and they have two flagella. The female sex organ is called archegonium, and it is flask shaped and produced a single egg. So this is the particular hanging archegonia you can see specifically. And here it is a male gametophyte, and in male gametophyte you can see the antherozoid. This is a female gametophyte, and hanging archegonia you can see here. Okay, so we are going to, this is the diagram of Markensia. We are going to discuss. I am just briefing you with the bryophytes. We are going to discuss the life cycle. So the antheridium is a male sex organ, archegonium is a female sex organ, and anthrozoids are the male gamete which are biflagellated, which are produced by the antheridium and are released into water, and they come in contact with the archegonium. An anthrozoid fuse with the egg to produce a zygote. And this fertilization is called zoidogamy, because here female gamete is large and sessile or non-motile, whereas male gamete is motile, and the fusion is occurring through the water. Because of this, it is known as zoidogamy. And here, zygotes do not undergo reductional division immediately, but it undergo mitosis and produce the embryo. Okay, so these are some of the preliminary points. Now we have to discuss the life cycle in detail. So what we have studied in case of general characteristic, they are the first land plant, but they are not successful land plant because they do not have a true root stem leaf. They originated in aquatic and then came on the water. So they need water for fertilization. Because of this, they are known as amphibians of plant kingdom, and they do not bear a vascular tissue. So we can call him as a non-vascular amphibians of plant kingdom. And they have a thyroid structure, leaf-like, root-like, and stem-like structures are known, and they are also known as zyophytes because they prefer to grow in moist and shady places. Now we are going to discuss here the life cycle. of this bryophytes. Now, first, before studying the life cycle, if we talk about the life cycle, in these two stages are found. One is gametophyte and one is sporophyte. If we talk about gametophyte, gametophyte is a gamete producing body and it is haploid. Whereas if we talk about sporophyte, it is spore producing body and it is diploid. That is 2n, whereas it is n. And this is a dominant phase of the life cycle. So it is a dominant phase, so it is independent. It is independent. Whereas this is dependent or parasitic on gametophyte. Okay. So this is gametophyte is independent and sporophyte is dependent, and it is dependent or parasitic on the gametophyte for the different purposes. Now it can be parasitic completely or it can be partially parasitic on the gametophyte. Okay, this gametophyte bear sex organ like antheridium and archegonium are present on the gametophyte. Whereas this sporophyte is differentiated into three parts, and that is differentiated into food, seta, and capsule. And that is the difference between gametophyte and. Now we are going to discuss the life cycle, and before studying the life cycle in detail, we have to know the structure of the sex organs, that is antheridium and archegonium, which are going to be present on the gametophyte. So if we talk about the structure of antheridium and archegonium, first we talk about the structure of antheridium. This is the structure of antheridium, a male sex organ. And in this male sex organ, you will see this is a stalk. This is a jacket because jacketed sex organs are found. These are the cap cells or cover cells. And this all cells which are found here, they are known as they are known as androcyte. So they are androcyte. They are known as androcyte or anthrozoid mother cells. Now it is known that antheridium is present on the gametophyte and gametophyte is haploid. So antheridium is going to be haploid. Means stalk, jacket cells or androcyte and anthrozoid mother cells are going to be haploid. And this androcyte are the anthrozoid mother cell or male gamete mother cell that undergo mitosis. Actually, that undergo mitosis and produce biflagellated male gamete. Okay. So when I will make a complete diagram of a life cycle, I will not draw this again. I will write just androcyte and the biflagellated male gamete. Then at that time you should understand. And this biflagellated male gamete look like this. You can see. Whereas if we talk about a structure of archegonium, the structure of archegonium is somewhat look like this. And these are the cover or capsules or lead cells. This is known as neck. This is cross section. Okay. And this part is known as venter. And here you will find this structure that is known as stalk. Now in neck, you will find some cells like this, which are known as NCC. That is neck canal cell. And here you will find VCC and egg cell. Egg cell is also known as oosphere, and VCC is ventral canal cell. Now this archegonium is also present on the gametophyte structure, so it is haploid. So cover cells or cap cells or lid cells are also haploid. Neck is also haploid. Venter is also haploid. Egg is also haploid. NCC is also haploid. VCC is also haploid, and stalk is also haploid. So all the structures are haploid because they are particularly what present on the gametophyte. So now, when you get clear with the structure of male sex organ and female sex organ, antheridium, we know that it is a male sex organ, and archegonium is a female sex organ. And when we get clear through this, both male and female sex organ, we can draw a schematic diagram of life cycle in a proper way. Now I am going to draw the life cycle properly, and then we will write some of the important points on that. So in life cycle, you know that gametophyte is the dominant phase. So I am writing gametophyte here as a dominant phase. Now, just we have discussed that gametophyte is haploid and it bear or has male sex organ and female sex organ. Male sex organ is known as antheridium and female sex organ is known as archegonium. So this antheridium is also haploid and archegonium is haploid. Now antheridium has antherozoid mother cell which is haploid. And this antherozoid mother cell undergo mitosis and produce antherozoid. But these antherozoids are biflagellated as we have discussed. So these are also haploid because they are produced through mitosis. Now archegonium has egg or we can say egg mother cell. That egg mother cell undergo mitosis and produce egg or oosphere which is haploid. Now this egg or oosphere undergo fertilization with the male gamete or antherozoid biflagellated. It is known as syngamy or fertilization, that is zoidogamous as we have discussed, and it produces zygote. So this male gamete is haploid, female gamete is haploid, egg cell is haploid, so the fusing zygote will be diploid. Now zygote undergo mitosis and due to mitosis it forms embryo. So several mitoses will occur. In a group of diploid cells will be observed that is known as embryo. Now this embryo will undergo mitosis and produce sporophyte. And that sporophyte is obtained, so it is diploid. Now see, the embryo is not forming in algae, but it is forming first time in case of the bryophytes. So the first embryophytes are no one other than bryophyte. Okay. Now sporophyte is differentiated into food seta and capsule, as we have discussed. It is differentiated into food. Sorry. Just a minute. This sporophyte is differentiated into three parts. That is capsule, seta, and food. So if sporophyte is diploid, this all structure are going to be diploid. Now capsule has spore mother cell which is diploid and that undergo meiosis and produce spore that is haploid. Now spore germinate and form a gametophyte. Means spore grow or divide or germinate and form a gametophytic body, which is haploid. So it is forming from the spore, because of this it is haploid. Now if we see, the diploid state is starting from here and here completes here and already started from here. So this is representing gametophytic generation. So all the stages which are included here, they are particularly associated with the gametophyte or gametophytic generation, whereas this are associated with the sporophyte, so they belong to sporophytic generation. Okay, so this is the thing where a life cycle occurs. So if you want to remember, what you need to remember? You need to remember just 2 to 3 things so you can remember the whole life cycle. You have to remember that which one is dominant, gametophyte is dominant, which one is dependent, sporophyte is dependent. So dominant is gametophyte, so obviously sex organ are present on gametophyte. So the sex organ will be haploid, so gametes will be also haploid. So gametes are produced by the mitosis. Gametes are anthrozoid, bifurcated anthrozoid, that is male gamete, and axil, that is female gamete. They fuse and form a zygote, that is diploid. And then after mitosis occurs, first embryophyte, so embryo will form, embryo will undergo mitosis, sporophyte will form. Sporophyte is a food cell capsule, so it has diploid structure. Capsule has a spore mother cell, which undergo meiosis and produce spore. And spore is haploid, so it will again germinate in form of gametophyte. Now here we remember this zygote is also known as oospore. So it is oospore and this is oosphere. So oosphere is unfertilized egg, so it is haploid. And oospore is fertilized egg, so it is a diploid. Okay, so this is the life cycle. Now what is remaining to understand in this part? That this has a diploid phases as well and haploid phases as well, but haploid phases or gametophytic phases are more dominant. So the life cycle is known as what? Haplodiploidy. The life cycle is named as haplodiploidy. But the spore is germinating and forming a gametophyte, and this spore germination occurs in 2 ways. The spore germination occurs in 2 ways. So next we are going to talk about the germination, spore germination. It is of 2 types. Direct germination and indirect germination. Now in direct germination, what happens? Spore directly form gametophyte. Spore directly form gametophyte without any intermediate stage. Intermediate or middle stage or phase. And this direct germination happens in case of liverworts and hornworts. These are the 2 groups of bryophytes. But in mosses, this indirect germination occurs. And in indirect germination, spore germinate to form multicellular filamentous structure known as protonema. Stage. This protonema stage is found, and on this birds are formed. And this bird develop to form gametophyte. So this is an indirect way of germination through the one middle stage or intermediate stage, that is filamentous, that is known as protonema stage. And this indirect germination happen in case of mosses. And it is known that this indirect germination is best for the. Okay, so this is the particular spore germination. So now I hope you have understood the life cycle completely in detail. So this life cycle is haplodiploidy. So we need to note down some of the points which we have understood from the life cycle. So what we have understood from the life cycle? First thing, that sexual reproduction is oogamous in case of bryophytes, and sexual reproduction is oogamous as well as life cycle is haplodiploidic. Third, what is unique character of bryophytes? So unique character is dependence of sporophyte on gametophyte. Okay. Then fourth, bryophytes are homosporous, means they produce only one type of spore, and they have a biflagellated male gamete and jacketed sex organ. That all are the important points that we have understood from the life cycle. Now we are going to talk about the classification. Okay, so I hope you have understood this life cycle in detail in a proper way, and what are mosses, that also you have understood. Now we are going to talk about the part that is classification. And before classification, here's some point which we have understood already in life cycle, that again we will revise. Multicellular body is sporophyte. Sporophyte is not free, as we have discussed. It is dependent on the gametophyte, and gametophyte is photosynthetic itself because it is free living, and sporophyte get nourishment from it. You can see in this diagram again. This is a gametophyte. These are the rhizoids, fruitlates, and this is a sporophyte. And sporophyte is dependent on this gametophyte. Okay. Now, some cells of the sporophyte called a spore mother cell undergo reduction division produce spore. The spore germinated from a gametophyte. That also we have discussed. All bryophytes are homosporous. That also we have noted down. They show heteromorphic alteration of generation because gametophytic and sporophytic bodies are conspicuous, means visible differently, and the life cycle is haplodiploidy. Okay, so we have discussed already life cycle in detail. Bryophytes in general are relatively of little economic importance. Some mosses provide food for herbaceous mammals, birds, and other animals that we will discuss. So we will discuss this partner point when we will talk about the mosses. So now let's talk about the classification of bryophytes. And bryophytes are divided into three classes, like Hepatocopsida, in which liverworts are included, Anthoceropsida, in which hotworts are included, and the Bryopsida, in which mosses are included. This Bryopsida is also known as mussy. Sometimes the mussy word is used for the Bryopsida. Okay, so we are going to discuss all these three classes in detail one by one. And out of these three, these two are of much importance according to Neet. This is important, but majority questions are being asked from the liverworts and mosses. So first we are going to talk about the liverworts. In liverworts, it is known that The name is given based on what? Their shape. And their shape is somewhat liver-like. Because of this, they are known as liverbirds. Now, if we talk about the liverbirds' habitat, they grow usually in moist, shady places, such as banks of stream, marshy ground, damp soil, bark of tree, and deep woods. These are the different habitat over which the liverbirds are known to grow. So you can see this is the conditions where the liverbirds can grow. When we talk about the plant body of liverbirds, bryophytes are thalloid. They have a root-like, stem-like, leaf-like structure. So liverbirds are also thalloid. And if we talk about their examples, the Margantia is there. You can see this is the diagram of Margantia, which is liverbirds. And apart from Margantia, several other examples are also known, like Porella, Pelia, Rixia. These all are known apart from the Margantia in liverbirds. When we talk about the thallus of this liverbirds, it is prostrate, means it grows horizontally. It is dorsiventral, means dorsal and ventral surface are there, and this dorsal ventral cell are closely oppressed to the substructure. Means this dorsiventral surface are close to each other. It is a flat body and it is closely oppressed to the soil or the substratum where they are found. And in this liverbirds, if we talk about the leafy member, which has a leaf-like structure or leaf, that leafy member is nothing but a porella. And this porella, leafy member is porella. And this porella has a tiny leaf-like appendages present on two rows on the stem-like structure. Means if I am growing, this is a stem-like structure. So the leaf-like appendages are present in two rows like this. Okay, so the porella look like this. Now we talk about some of the more important points. If we talk about this bryophytes or liverbirds in more detail, then we come to know that they have a dorsiventral and thickened body. They have a thalloid body, and in thalloid body also they have rhizoids. And in liverbirds, it is known that rhizoids are unicellular and unbranched. Apart from this, here in liverbirds, it is known that sporophyte is completely dependent or parasitic on gametophyte. Why it is completely dependent or parasitic? Because it depends for food, water, and habitat. Okay, so that point you should remember. Apart from this, the one of the known important example for oldest liverbird, if we talk, the Rixia is primitive liverbird, and its sporophyte has capsule only, means it is not much differentiated. Okay, and this rixia are flat. Now, the one of the important member on which the name liverwort is given to this class is Markensia, which is liver-like. So, we are going to discuss Markensia in detail. So, let's talk about Markensia, which is one of the important member of this liverworts. Now, Markensia, if we talk, it is a dorsi-ventrally flattened and dichotomously branched. Dichotomously branched means, suppose this is the branch, it will divide into two, and further also it will divide into two. So, like this division occurs, because of this, it is known as dichotomously branched. Now, in liverworts, this is a dichotomously branched. Markensia is same, and this Markensia is dioecious. One of the important points here, dioecious means male thallus and female thallus are different. Now, if we draw the male thallus, then male thallus look like this. This is midrib. Here you will see the presence of rhizoids. And here you will see the presence of this cup-like structure, which are known as gamma cup. And over this notch, you will see the presence of elongated structure, which is known as anthradiophore. And over this anthradiophore, you will see 1, 2, 3, 4, 5, 6, 7, 3, 4, 5, 6, 7, 8, 8. So this is 8-lobed receptacle. And this is anthradiophore. 4 means stalk, over which anthridium are present in this receptacle. And this anthridium, as we have studied, produce the male gamete or anthrozoid, which are biplanetated. So this is the structure of male thallus. And apart from this, next we are going to talk about female thallus. Female thallus also look like somewhat this, mostly similar. But here in place of anthradiophore, there is an archegonium. And over this archegonium, you will find receptacle with the 9 cylindrical layers. 1, 2, 3, 4, 5, 6, 7, 8, 9. This is a receptacle, has 9 cylindrical rays. So this is female thallus. So the male thallus and female thallus can be differentiated only by the receptacle. Okay. Now further what happens? If we see the one cylindrical ray, structure of cylindrical ray, you will find hanging archegonium. And this hanging archegonia is present here in each ray. And this hanging archegonia has an egg cell, as we have discussed. So what happens? This antheridium produce a male gamete. Through water, they reach to the neck of archegonia. Then fertilization occurs here. And after fertilization, here zygote will be formed. Zygote will develop into embryo, and embryo will develop into sporophyte. Because of this, sporophyte is going to develop on this. So this sporophyte is dependent on this gametophytic body, that is thallus. Now this is thallus, so this all are going to be haploid structure because this is gametophyte. And after fusion, particularly we can say the sporophyte will develop. So this is the gametophytic structure in case of Markensia. How the male and female gamete are produced, where they are located, that we have discussed. Now after fertilization, embryo will form, and embryo after embryo, what will form? The sporophyte will form. So now after this, we will be going to deal with the structure of sporophyte. But before studying sporophyte, we should understand what are gametes. So if we talk about gametes, gamma cup produced. Gamma cup are the small receptacle. They are the small receptacle located on thallus. But this gamma cup produce gamete, and this gamete, if we talk, the singular is gamma, are green multicellular asexual bud. They are asexual bud. They are haploid. And this green multicellular asexual bud detached from parent body detached from parent body and germinate and give rise to two new thallus. Or thalli. Now after, so this is what a gamete comes. So one gamete can give rise to the two new thallus. That you need to remember. So if 20 gamete are there, then they can produce what? 40 new thallus. So that is the one of the important speciality of this gamete. And this Markensia we have discussed, that is a dioecious. Male and female sexual reproduce are the different thallus. Whereas the Rixia which we have discussed, that is a monoecious. The male and female sexual are of the same plant. Now we are discussing now Markensia. So in Markensia, gamma we have discussed. We have discussed the structure of thallus. Now, after fertilization, the zygote will develop into embryo, and embryo will develop into sporophyte. So now we are remaining with the structure of sporophyte in liver buds. So if we talk about the structure of Sporophyte. Then it is known that suppose this is the hanging archegonia, and we have studied that just now that over hanging archegonia, this is a cylindrical ray, and below which hanging archegonia is found, where egg cell is found, where zygote is found, where embryo is formed, and that embryo develops this sporophyte. So here the foot is formed first. This is the elongated structure which is known as theta. So this is foot, this is theta, and below which you will find the presence or occurrence of the capsule. This is whole sporophyte, and in this sporophyte, this region is known as foot, this region is known as theta, and this is known as capsule. These all are diploid. And in capsule, we can see the presence of spore and the true letters. So these are elaters, and these are spore. Spore are haploid, but elaters are diploid. And these elaters are the true elaters with spiral thickening. As you can see in these elaters, the spiral thickening is found here, and that elaters help in spore dispersal. So this spore will be released from this capsule after its breaking, and this spore will reach to the suitable substratum, and then it will form a new gametophytic body under the sexual reproduction. So this whole structure is nothing but a sporophyte. Okay, I hope you are now clear with the Marquisia structure and clear with the liverworts. Now after this, we will be talking about the hormones. So vegetative reproduction in liverworts takes place by the fragmentation of thalli, that is also important, or by a specialized structure called a gamete. So gamete is a means of vegetative reproduction. We have discussed. See, this is the gamete, how they are produced in the particular structure or thallus of the liverworts. Then gamete we have discussed being multicellular asexual bud which develop into small receptor called gamma cup. Okay, and this gives rise to these are the gamma cup. You can see the gamete detached from parent body. We have discussed this. During sexual reproduction, male antheridia and female archegonia sex organ are produced on the same or different thalli. This is the archegonia. You can see this is the antheridia. The sporophyte is differentiated to the food seta capsule. We have discussed, and this is the diagram of meiosis. We will discuss that later on. It is not of liverworts, and spore will be produced within the capsule. That spore germinate in form a new gametophyte. Some members of members like Marquisia produce elaters in the capsule, which have been spore dispersal. That also we have discussed. spore germinate to form a free living gametophyte. Okay, so now we are going to talk about hornworts. That is the second rule. Okay, dear student, I hope you are clear, you are getting the picture. You are now able to understand or differentiate between algae and bryophyte. Now we are going to talk about the second class, that is hornworts, that are known as Anthoceropsida. Now in hornworts, the plant body of hornwort like Anthoceros is also thalloid, and it also consists of rhizoid. It is a root-like structure. You can see this is the Anthoceros. Now they possess elongated horn-like sporophyte. Because of this, they are named as hornworts. See, the sporophyte is horn-like, so they are named as hornworts. And the sporophyte consists of foot, intercalary meristematic zone, and a capsule. Three structures are found. Means seta is absent. Foot, seta, capsule. But here, foot and capsule are found. Here, in case of hornworts, specifically, seta is absent, and in place of seta, meristematic zone is found. The capsule produce spore and pseudoelaters. In this, pseudoelaters are found, and pseudoelaters are the elaters where spiral thickening is absent, and this also helps in spore dispersal. See, these are the pseudoelaters which are helping in spore dispersal, and spore are released in mature capsule like this. So, what do you mean by pseudoelaters? They lack spiral thickening, but function remains same. Okay. Then, if you talk about this hornworts, then they have a root-like, stem-like, leaf-like structure, and they have rhizoid, but rhizoid are unicellular and unbranched. The branching is not found in case of rhizoid. And in hornworts, sporophyte is not completely dependent. It is partially dependent. Gametophyte. It is partially dependent on gametophyte because sporophyte is photosynthetic. They can manufacture their own food. So it depends only for water and habitat, not for food. Because of this, it is known as partial parasite or semi-parasite. So means these hornworts are partial parasite or semi-parasite. In this, in place of seta, meristem is present, we have discussed. And due to the activeness of this meristem, capsule grows rapidly and that grow like a horn. Because of this, they are hornworts. Now, one more important point that we are going to discuss, that hornworts or anthoceros represent algal ancestry, specific green algae ancestry. Means hornworts are evolved or developed from the green algae, because hornworts have pyrenoids in chloroplast. And we have studied in algae also, green algae also, there is also a presence of pyrenoids. And in hornworts also there is a presence of pyrenoids. So if we link this thing, then we come to know that hornworts, the ancestors of hornworts are nothing but green algae. Okay, so this is all the important points or the information that is necessary with respect to Anthocerosida or hornworts. Now, after this, we are going to discuss Bryopsida. That is mosses. If we talk about the Bryopsida or mosses, the predominant stage of the life cycle of moss is the gametophyte, and this gametophyte consists of two stages. The first stage in gametophytic body or dominant body of mosses, which is seen, that is juvenile stage, and it is called a protonema, which develop directly from the spore. Like this, you can see. So this protonema is somewhat filamentous, creeping, and branched structure. But later on, the second stage is developed in the life cycle of mosses after the first stage, which is developed from the spore directly. The second stage, after the protonema stage, that develops, that is the adult leafy stage, and it is called a gametophore, which develop from the protonema as a lateral bud, and this bud grow and form a developed gametophyte. Okay, so this is the some points with respect to mosses. So in mosses, two stages are known, that is primary stage, protonema stage, and second stage, leafy stage. Now, if we talk about general character of mosses, in mosses also there is a stem-like, root-like, and leaf-like structure, but they have a rhizoid, and rhizoid in case of mosses are multicellular and branched with oblique septa. Suppose this is the rhizoid. This can be branched like this, and this oblique septa are formed. Okay. Now, if we talk about the mosses, the vegetative reproduction in mosses occur by fragmentation and budding in secondary protonema. But during sexual reproduction, sex organ are produced. At the apex of leafy shoot. Now, if we want to discuss the sporophyte of this mosses in a more detail, then we have to understand the funaria, that is one of the important examples in case of the mosses. But before studying the detailed structure of this funaria, we have to talk about nutrition. And the mosses are also partial parasite or semi-parasite, because Sporophyte depend on gametophyte only for water and habitat because sporophyte is photosynthetic itself. Now, one statement is that, which is the important one that I am going to write here. I will discuss how this statement is important in the detailed part of Funaria. See, the sporophyte of mosses is more elaborate or developed than liverworts. As we go in the evolution, as we talk about the evolution, the sporophyte become larger or developed and gametophyte become reduced. Okay, it is the evolution. So compared to algae in bryophyte, the sporophyte is developed. And in bryophyte also, after liverworts, the mosses has evolved. So in mosses, the sporophyte is elaborate or developed than the liverworts. And one more important point that the mosses have elaborate mechanism of spore dispersal. Now, what does this statement mean? That I will show you in now the coming slides while discussing the Funaria in detail. So let's talk about the example of mosses first. And this in this example, if we talk of mosses, then one of the important example is Funaria. Then second example is Polytrichum. The third example is Sparna. And we are going to discuss about these three in detail. First, we will discuss the Funaria. If we talk about Funaria, the Funaria is monoecious. Actually, what happens when Funaria develops initially, it develops one male branch. And over this male branch, they have some leaf. And then it develops a female branch. But female branch grows slowly, and it overtops the male branch. So in female branch, they have a presence of archegonia here. And this is a male branch. And they have an amphidium at this position. So just we have discussed in mosses initially that the male and female sex organ are present at the apices or apex of the leafy shoot. So this is the amphidium, that is male sex organ. This is archegonium, that is a female sex organ. Now male gamete reaches from here to here, and fertilization happens, and then this will develop into what? A sporophyte. So when sporophyte develops from zygote here, it looks like this. And if that fruit remain inserted within this leafy part, seta is here, and this is capsule. So this is the sporophytic body, and this is gametophytic body. Okay, this is haploid body, this is diploid body. Now the capsule has a spore mother cell. Now the labeling of this should be remained, okay? Because in NCRT, the labeling which is given, they usually ask a question from this main axis, and this is rhizoids. Okay, so this is the thing of the particular funaria. Now in funaria, the capsule has a spore mother cell which undergo meiosis and produces spore. So capsule has a one detailed structure. Capsule is also divided into three parts in case of funaria, and then it will take part in the spore dispersion. So if we draw the structure of capsule in case of funaria, it has a three parts, and it looks like somewhat this. The structure is somewhat like this. And in that structure, so this is the structure of what capsule. In this region is named as operculum. That is the first part, and this is diploid because it is a part of capsule. And in that you will find this structure that is known as peristomial teeth. Okay, so these are also diploid. Now here, these are the spores. This is one structure in between, that is columella. These are the spore, and these spore are produced within spore sac. So this second structure is known as theca, and this third region of capsule is known as apophysis. So now this spore are present in spore sac, and in between there is a columella, and here peristomial teeth. So here what happens? This columella get dried, so the space get formed between this, and this polar spore get released from here, but that are dispersed by peristomial teeth. So peristomial teeth help in spore dispersion. So this difficult mechanism, complex mechanism is seen in case of this complex mechanism is seen in case of the capsule of funaria. Because of this, and peristomial tip help this complex way occur for the spore dispersal also. So because of this, we call it as an elaborate mechanism of spore dispersal is found in case of this funaria. And in this funaria or mosses, one more important point you should remember that elaters are absent. Now after this funaria, we are going to talk about we are going to talk about the remaining examples, that is polytrichum and spartum. We remember funaria is also known as cord moss or rock moss. Funaria, also known as root moss or cord moss. Okay, so this is about the moss, that is funaria. Now we talk about a sphagnum. Okay. So the mosses have elaborate mechanism of spore dispersal and contain a peristomial teeth for the dispersal of spore that we have discussed. And the C, like this, the peristomial teeth are seen in case of the structure. This is theca, this is epiphysis, and this is peristomial teeth. Now, after this, we are going to discuss about the another example, that is sphagnum. Now, if we talk about sphagnum, it is known as pit moss. Why it is known as pit moss? Because pit formation take place by fossilization of sphagnum. So fossilization of sphagnum will occur, and it is known that this sphagnum grow in acidic marshes. This sphagnum grow in acidic marshes, acidic areas. So what happens in bog? The number of bacteria are less. So due to which the degradation of the dead cells of this sphagnum does not take place. Hence, it is found or present in the form of fossil. So the fossilization is occurring. Apart from this, so it is also used as fuel. One more thing, they are also known as, sphagnum is also known as absorbent cotton or cotton moss. Now, why it is known as absorbent cotton or cotton moss? Because sphagnum absorb water in very high amount. Therefore, it is used as absorbent cotton in the Europe countries. Apart from this, it is known that this sphagnum help in transportation of cut plant parts. Okay, so these are the something with respect to the polytrichum and this about the sphagnum. Okay, now after this we are going to talk about another example, that is polytrichum. This polytrichum is known as hair cap moss, and in polytrichum one species is there that is known as Polytrichum commune. It is used in kidney stone removing. Now, apart from this example, it is known that mosses, actually bryophytes in general, if you talk, they are of little economic importance, but mosses provide food for herbaceous mammals, birds, and other animals. Apart from this, it is known that mosses along with lichen, means after lichen, are the first organism to colonize rock. Hence, they are of great economic importance. So they are of great economic importance. Now, apart from this, it is also known that it is also one of the more importance of mosses is that mosses form dense mat on soil. So they reduced impact of falling rain and prevent soil erosion. So mosses also play a role in prevention of the soil erosion. So that is about the mosses, okay, and their role. So common example of mosses we have discussed already: Funaria, Polytrichum, and Sphagnum. These are some of the examples. This is a haircop moss. This is a peat moss. Okay, and let's talk some of the few MCQs. Easiest MCQ. Amphibians of plant kingdom are, you know that they are bryophytes. Then mosses possess a root-like structure which are known as rhizoid, and we have studied they can be unicellular or multicellular, they can be branched or unbranched. All bryophytes are homosporous, we have discussed already. And the common name of Funaria is cordmos. Okay, so this is all about what a bryophyte. So in a bryophyte, we have discussed in detail about the general character, their life cycle, structure of sex organs, that is Antheridium and Archegonium, then their classes in detail, like liverworts, onwards, and mosses. So I hope you understood it in a very well manner or in detail. So thank you. In next class, we will be continuing with the